Okay, so we've had a chance to look at the nitty-gritty details of cellular respiration, aerobic cellular respiration, looking at the details of glycolysis and then pyruvate oxidation, the Krebs cycle, electron transport chain, and chemiosmosis. So now what I'd like to do is bring us back together and look at and give a brief summary of the net gain of ATP per glucose molecule. So first of all, we know that you may not have really been keeping count, but the theoretical yield of cellular respiration is 38 ATP, and that's 38 ATP molecules per one glucose molecule. The actual yield, we know in science, and particularly in experiments that we've done, where the actual yield is typically less than the expected or theoretical yield. And, well, in an experiment, we would say that can be attributed to various sources of error. In the case of ATP production, really, we can think of our cell as one big experiment. So what is not happening in an ideal fashion? Well, let's think about that. So take a minute to pause the video and think about why that might be. Why might the actual yield be less than the theoretical yield? Okay, well, here are some reasons. Okay, the first reason is that the inner mitochondrial membrane is in fact not completely impermeable to hydrogen ions. And remember that we're relying on that concentration gradient existing and for those hydrogen ions specifically to go through ATPase in order to create that proton motive force. And that's going to enable our ADP molecules to be phosphorylated into ATP. So if our hydrogen ions are in fact not going through ATPase, we are not ending up with any ATP produced from that hydrogen ion. So that's one piece of the puzzle. The second piece, right, so there we go. Some are leaking through without passing through ATPase. The second is that some of the hydrogen ions that might be in that intermembrane space might get used up for things that we don't, that we wouldn't, think of. And so therefore, they're also not passing through ATPase and therefore not producing any ATP. That's not unlike sort of side reactions in a chemistry experiment. So I thought it'd be helpful for us to discuss, to do a breakdown of those 38 ATPs and how the theoretical yield is 38 ATPs. Where are they coming from, right? So I want to differentiate between the ATP that are produced via substrate level phosphorylation and oxidative phosphorylation. Remember, substrate level phosphorylation is happening directly and during that particular stage, whereas oxidative phosphorylation, the electron carriers are produced, like for instance NADH and FADH2, they are produced in a particular stage, but the phosphorylation and ATP production is happening only during the electron transport chain and chemiosmosis during those final stages. All right, so let's take a look at this. So first of all, in glycolysis itself produces 2 ATP. You'll remember that we invested 2 ATP, but got 4 out, so that's a net gain of 2 ATP, and that's via substrate level phosphorylation. The 2 NADH that are produced during glycolysis, you'll remember that each NADH, so I have 2 NADHs, but those are each pumping out 3 hydrogen ions into that intermembrane space. And we know that that's going to yield 6 hydrogen ions available to pass through ATPase, and thus 6 ATP, right? So 3 hydrogen ions per NADH gives us 6 ATP in total, and that is by oxidative phosphorylation. Next, pyruvate oxidation itself. You'll remember that I have 2 pyruvate molecules. So I'm going to write 2 pyruvate, pyr for short. And then, so each of those pyruvates is going to produce an NADH, right? So if each NADH, again, is going to have, so each pyruvate is producing one NADH, and each NADH is pumping 3 hydrogens into that intermembrane space. And so that's going to give me a total of 6 hydrogen ions, which will become 6 ATP via oxidative phosphorylation. Okay, the Krebs cycle. So you'll remember that in the Krebs cycle, we produced 2 ATP, but first they were GTPs, right? But we need to remember that the Krebs cycle, we look at per acetyl coenzyme A that enters the cycle. So if we have two pyruvates, we're going to produce 2 acetyl coenzyme A's, and thus 2 turns of the Krebs cycle per 1 glucose molecule. So if for 1 turn of the Krebs cycle we produce 1 ATP, 2 turns are going to give us our 2 ATP, and that's happening directly. So that part is happening via substrate level phosphorylation. Now let's talk about the next part, and the next part are these 6 NADHs that are produced. Well, you'll remember that 1 turn of the Krebs cycle produces 3 NADHs, so 2 turns are going to produce 6 NADHs. So it might be helpful for us to write in that this is 2 turns of the Krebs cycle. So, and both of these right here, because they are electron carriers, those are both oxidative phosphorylation. That's the mechanism of ATP production. So with our 6 NADHs, we have 3 NADHs times 2 turns. I'm going to write 2 turns. And we know that that's going to give us our 6 NADHs. And then we're going to say times 3 H pluses per NADH is going to give us 18 ATP. Now, same kind of dealio with the FADH2. We know that there are 2 FADH2s produced, but that's via 2 turns of the Krebs cycle. So per turn of the Krebs cycle, we have 1 FADH2. So 1 FADH2 is produced per turn. And that will give us 2 FADH2s. But remember that for each FADH2, we're only able to pump out 2 hydrogen ions because it is a little bit more electronegative. It's not going to give up its electrons to NADH dehydrogenase. It's going to give it up to our next our next protein complex in the electron transport chain. So that hence the 2 protons, and those 2 protons will translate into 2 ATP, but because we have 2 FADH2s times 2 protons pumped, that will give us 4 ATP in total. So adding all of that up, we can see 2 plus 2 plus 6 plus 6 plus 2 plus 18 plus 4 is going to give us our theoretical yield of 38 ATP. So I hope that made sense, and if there are any points that you need clarified, please let me know.